all these people you're seeing here have very different diseases. For example, one of them is suffering from Alzheimer's disease, keeps forgetting things and is often confused and without orientation. Another person has gout, which causes a lot of pain and swelling in the joints and the inability to move them properly. Yet another person has atherosclerosis, which puts them at risk for a heart attack or a stroke as a result of blocked blood vessels causing insufficient oxygen supply of the heart or the brain. Even though these diseases look so different from each other, they have something in common. In all diseases, an immune protein called NLRP3 is activated. For Alzheimer's disease, the activation occurs in the brain. For gout, it happens directly in the joints. And for atherosclerosis, NLRP3 plays a role in the walls of the blood vessels within the whole body. But how does NLRP3 get activated? What happens in the tissues? To give an example, let's look closer at the person suffering from atherosclerosis. Here you can see what a blood vessel of a healthy person usually looks like. However, during atherosclerosis development, lipid-carrying lipoproteins and lipids like cholesterol, which are taken up by consumption of fatty meals, accumulate in the blood vessels of the body. Cholesterol is usually transported through the body in a soluble form, but when there is too much cholesterol present, it might accumulate in various tissues and form solid crystals. And this is exactly what happens in the wall of blood vessels during atherosclerosis. And this accumulation of lipids and crystalline cholesterol is then recognized by circulating immune cells, which enter the tissue where they try to remove these abnormal deposits. This results in the thickening of the vessel wall and the formation of so-called atherosclerotic plaques. When the immune cells cannot entirely clear the cholesterol crystal deposits, they activate a warning system to call other immune cells for help. And this warning system involves the activation of our immune protein NLRP3, which we'll look at in more detail in a moment. However, the recruitment of more and more immune cells causes the growth of the atherosclerotic plaques during disease progression, and consequently the vessel lumen becomes narrower while the vessel wall becomes unstable. Eventually the plaque ruptures, which results in the formation of a thrombus that clogs the vessel. Hence the surrounding tissue cannot be supplied with oxygen, and that's what happens when a person has a stroke or a heart attack. To be able to prevent or attenuate this process, we need to understand what is happening on the cellular level. Amongst the innate immune cells in the atherosclerotic plaques, there are many macrophages. Their name is Greek and means big eaters, and that's exactly what they do. They try to eat up and remove things that are not supposed to be there, such as pathogens or also cholesterol crystals. So the macrophages try to eat the pathogens or the crystals in a process that is called phagocytosis. They surround the foreign objects with some of their membrane to form a phagosome, which gets taken up into the cell. This phagosome then fuses with a lysosome, which has a low pH and enzymes to degrade anything the macrophage has phagocytosed. For many things this process works well, but crystals cannot be digested properly. The macrophage just does not have the tools. The macrophage, however, just keeps taking up crystals, until finally one of the phagolysosomes ruptures. And this is a signal for the cell that its capacity has been reached and it has lost its function. For the cells, it is very important to keep the contents of the lysosomes contained so that the enzymes don't get released into the cytoplasm and digest the cell itself. And this is where our protein of interest, NLRP3, comes into play. NLRP3 is located in the cytoplasm of the macrophages, and under homeostatic conditions, it is in an inactive state. While it is not completely understood what exactly is sensed by NLRP3, it is known that it recognizes a wide range of pathogens, but also cell damage, such as the disruption of the phagolysosome compartment after crystal uptake. So once NLRP3 gets activated, it changes its conformation, which enables it to assemble the so-called inflammasome. Some clustering NLRP3 molecules function as a seed for inflammasome formation. But let us simplify this NLRP3 cluster to keep an overview. This NLRP3 cluster recruits an adapter protein called ASC. Actually, the entire ASC of the cell comes together within a single location within the cell, the so-called ASC spec. This polymerization is incredibly quick and happens within a few minutes. The ASC spec represents a big platform for downstream signaling. Procaspase-1, an inactive protease, is recruited to the ASC molecules, and this recruitment brings procaspase-1 into close proximity to each other, which results in the cleavage and the activation of the proteases. Active caspase-1 is then able to process pro-inflammatory cytokines, such as the potent fever-inducing interleukin-1 beta. Interleukin-1 beta exists in a non-active proform that requires the cleavage by caspase-1 to become activated. Moreover, caspase-1 activation also results in the death of the activated cell, which had lost its function anyways. During this form of cell death, called pyroptosis, pro-inflammatory cytokines are released to attract and activate other immune cells to deal with a persistent problem, for example, the cholesterol crystal deposits in the atherosclerotic plaques. The details of NLRP3 activation after the macrophage has lost its function are not quite understood, but there are several theories out there. One idea is that during the lysosomal rupture, enzymes get released into the cytoplasm that are usually not present there. These enzymes could modify a cellular protein, which might then serve as a ligand for NLRP3. Other theories suggest that ion channels or pore-forming toxins destabilize the ion gradients across the membrane, and that this triggers processes which are somehow recognized by NLRP3. For example, it is discussed that during NLRP3 activation, an efflux of potassium ions from the cytoplasm to the extracellular space occurs. A well-described receptor regulating potassium efflux is the so-called P2X7 receptor. Upon sensing of extracellular ATP that is released as a danger signal from dying cells, it releases potassium ions from the cell. Calcium ion mobilization might also play a role. One effect of increasing intracellular levels of calcium ions is mitochondrial stress, which can also be caused by enhanced phagocytosis or lysosomal damage. Stressed mitochondria, in turn, release reactive oxygen species, which also have been described to play a role in NLRP3 activation. However, we still don't understand how all these different pathways activate NLRP3. Likely, there is a yet unidentified step upstream of NLRP3 on which all of these signals converge and which leads to NLRP3 activation. So there are still lots of things we do not fully understand and need to investigate further. But why do we care? While we have focused on the activation of the NLRP3 inflammasome by cholesterol crystals in atherosclerosis so far, there are many other diseases where NLRP3 also plays a role. For example, in gout, there are uric acid crystals accumulating in the joints, which are also activating NLRP3. Similarly, in Alzheimer's disease, immune cells of the brain recognize amyloid plaques, and this elicits an immune response via NLRP3, which damages the neurons and results in memory loss. So if we improve our understanding of what happens during NLRP3 activation, we may hold the key for better therapies for all of these diseases. Blocking excessive NLRP3 activation could potentially help a lot of patients, and therefore we need to better understand the molecular details of inflammasome activation. And that's why we, at the Institute of Innate Immunity, within the Excellence Cluster Immunosensation at the University of Bonn, are working on unraveling the mysteries of NLRP3 activation and regulation.